Morning guys, day 3. So as promised, I said I was going to kind of finish up some stuff with DNA replication for you guys, and I wanted to go through just a quick review before we move in, and I actually look at the protein synthesis notes with you guys that I've asked you to kind of go through and copy into your note packets. So I'm going to do it hands-on for you guys. So on the table, you guys are going to see kind of a whole cell setup. We'll get to the second part in the next video, which will take place more or less tomorrow. But the white paper that you guys see here is going to be the nucleus within the cell. If you guys remember, the nucleus of the cell is where your DNA is located. It can't get out of the nucleus because it's double-stranded and it's too large to get out. So with this particular molecule, we said that it makes up the chromosome. So this chromosome is in a double helix. But there's an enzyme that's going to come through and kind of unwind it and break some hydrogen bonds for us, and that's going to be helicase. So my strand may look like that. And this may happen multiple places along that chromosome in order to get this process of replication complete. So we need to go through and make an identical copy of this strand as we grow, as we develop, as we repair, and we got to make new cells. This process is what has to happen. So when we looked at mitosis and meiosis, this is the S phase that we're looking at here. So thinking about that DNA molecule, if you guys remember, I have a sugar-phosphate backbone, and it will alternate sugars and phosphates all the way down. My sugars are the ones that are going to be attached to my nitrogen bases, and the nitrogen bases are making up the rungs of my ladder. Of those 4 nitrogen bases, adenine, thymine, guanine, and cytosine, 2 of them are larger. So you will notice that my adenines and my guanines are larger base pairs. They are called my purines. My smaller ones are cytosine and thymine. They are my pyrimidines. They are single ring structures. So I always pair together one purine with one pyrimidine. So I know that adenine or guanine and cytosine will pair together, as well as thymine and adenine. So adenine's a purine, thymine's a pyrimidine. I know which purines and pyrimidines go together because of hydrogen bonds. Guanine and cytosine will always have 3 hydrogen bonds between them. Thymine and adenine will always have 2 hydrogen bonds between them. If I look at a nucleotide, 1 sugar, 1 phosphate, 1 nitrogen base. So I have a whole bunch of these nucleotides joined together to make this nucleic acid. So like I said, helicase is going to come in and it's going to start to break those weak hydrogen bonds for me. What that does is it creates what is known as a replication fork. And I said there's really no set location because that replication fork is moving up as those bonds are breaking all the way through. And now what's going to happen is we have to read and make DNA in certain directions. So we talked about a 3' end and a 5' end on your DNA molecules. I said if it's not given to you, you need to go back and look at the deoxyribose sugar on your DNA strand, find the oxygen, and then count those carbons clockwise. Is it my 5th carbon that faces upward, or is it my 3rd carbon that faces upward? And that's how you guys will determine which ends. If you know one end of your DNA molecule, we said DNA is anti-parallel. So you'll see if this is 5', I know this is 3'. If this is a 3' end, opposite end has to be opposite as well, 5'. So helicase, like I said, has come in. It's broken apart those weak hydrogen bonds. So now we need to make and read our DNA. DNA polymerase is going to come in and do that for us. So DNA polymerase is going to hook up to my 3' end. It's reading DNA 3' to 5'. So this polymerase is working in this direction to go through and read. And he's reading, oh, I have an adenine. Oh, let me bring in a thymine. And he's going to start attaching new nucleotides, sugars and phosphates and nitrogen bases. So as it goes through, like I said, it's being read in this direction. DNA is now made my new strand. Since they're opposite and anti-parallel, 5' to 3'. So my blue strand is being made 5' end to 3' end. It is being read 3 to 5. So that's now my one new strand that I have there. On the other side, the same thing is happening. However, it's still being read 3' to 5'. It's going to get read opposite direction since they are anti-parallel. It's still being read 3 to 5. It's still getting made 5 to 3. So if my replication fork was only up to here to begin with, and it is being read 3 to 5, my base pairs are coming in, still polymerase. It's going to come in, it's going to attach, and it's going to put my new strand in. And it's going to be making it 5' to 3'. But then it gets to the end, and what it notices is it goes, oh, polymerase gets to the end, he kind of breaks off, and then he realizes that what happened is that the rest of the replication fork is opening up. Polymerase then has to go back up, and it has to kind of hook back up to the strand. In order to do that, RNA primase is going to come in and add a few little nucleotides, which is a docking point so polymerase can attach back onto the strand. Polymerase attaches back on, it starts making that strand once again, 5 to 3. If we remember, those strand, or that particular strand that we just created, is in fragments, and those are those Okazaki fragments. Since this strand is made away from the replication fork and has Okazaki fragments in it, this is my lagging strand. It is made away from the replication fork. And what happens is that these fragments, these Okazaki fragments that we see, are going to get glued together by my other enzyme called ligase. This strand over here is made continuous. It is made towards the replication fork. Because of that, that is now my leading strand. By the time I'm done, however, what you guys are going to notice is that those strands are completely identical. And we talked about semi-conservative replication. Each strand has 1/2 old DNA, which was my white, 1/2 new DNA, which is my blue strand. Completely identical all the way down if we look at those base pairs. Now my cell has twice the amount of DNA, so it kind of be like that diploid replicated stage that we were talking about. What has to happen is cell division, mitosis, meiosis. This cell is going to go through and divide. So by the time we're done, we now just have that one single strand in there again. So that is DNA replication. We'll continue with the rest of the process and what we're going to do with this strand as we work our way through protein synthesis notes.